works, great. Okay, so good afternoon, everyone. I'm Mike. Before I start, I just want to thank the organizers for putting together this fantastic conference and for giving me an invite to give a talk. I'm a postdoc in the labs of Beth Stevens, and I'm a Microsco at Harvard Medical School on the Broad. I'm going to tell you about my work developing a toolbox to model human microglial states in vitro. So, as we all know, microglia are the resident immune cells of the brain. They play critical roles in almost every aspect of brain function. And moreover, human genetics has implicated these cells in the development of multiple different diseases, including Alzheimer's, Parkinson's, and multiple sclerosis. And over the last 5 years, many studies have revealed that microglia do not exist as a homogeneous cell type, as many of the great talks before me have highlighted, but rather they can adopt subtypes called states. And these states have distinct gene expression in addition to a core microglial signature. And you can see that here in these single cell plots, maybe death by t-SNE rather than UMAP this time, where each dot is a microglia, and cells with similar gene expression cluster together into different states. So microglial states are found in the mouse and the human brain and have been discovered with a variety of different approaches, not just transcriptomics. And a striking feature of microglial states is that they are especially prominent and diverse during development, pathology, and repair, suggesting they might play central roles in these processes. And a key example of microglial states are, of course, the disease-associated microglia that we've heard so much about today already, which corral around amyloid beta plaques in Alzheimer's disease, but are also present in several other neurodegenerative disorders, and a similar state is also present in development, as we heard earlier from Bart. So DAM are thought to be protective in Alzheimer's disease, but beyond that, we know very little about their functions. And in fact, while we've been successful at cataloging different microglial states across different contexts, there are many unanswered questions about their biology. So firstly, the function of the majority of microglial states are completely unknown. Microglia play important roles, they're implicated in disease, but we don't have a clear sense of how that relates to these different microglial subtypes. Second, we don't understand how microglial states are established by transcription factors and other genetic regulators. And finally, what little we do know about the biology of microglial states is from mice. We don't really have any good models of human microglial states. So there's a clear need for new tools and platforms to study these microglial subtypes, but in particular to study those found in the human brain. So we set out to solve this problem using stem cell-derived microglia to build a platform to generate and manipulate human microglial states in vitro. And it was important for us to focus on scalable and tractable solutions, in part because microglial states can have hundreds of differentially expressed genes, and therefore we really want to be able to use high-throughput biology to interrogate their function. So in today's talk, I'll show you how we use stem cell-derived microglia to generate and manipulate human microglial states in vitro. This work has been recently published, and all the data is online and easily accessible. So to generate large numbers of human microglia, we use stem cells, a very similar protocol to what Jennifer described, although my cells don't look as nice, so maybe I need to talk to you, Jennifer, after. We differentiate these cells into induced microglia, which we call IMGLs, using a 40-day protocol from Matthew Blurton-Jones' lab, and we expose them sequentially to different growth factors and cytokines. And this approach produces cells highly similar to microglia in vivo, and these cells phagocytose, migrate to appropriate cues, and secrete cytokines. But our early work suggested there was a very limited degree of diversity or states in these cultures. And so we started to think about ways to increase microglial state diversity in vitro. So we know from prior work that subtype or microglial state diversity increases in response to injury or neurodegeneration. And so I reasoned that by exposing IMGLs to different substrates found in those contexts, perhaps we could induce different microglial states in a dish. So we selected 4 substrates that we thought might be representative of a degenerative context. We select synaptic debris, myelin debris, synthetic amyloid beta fibrils, and apoptotic neurons. And we exposed IMGLs to each of these substrates and performed single-cell RNA sequencing 24 hours later. And to my great delight, the first thing I noticed in this data set was a large degree of transcriptional diversity. So when I grouped cells into clusters, which you can see here in different colors on the left, I found 11 different microglial clusters in vitro. And you can see on the right here that this transcriptional diversity is driven mostly by exposure to the different CNS substrates, shown here in gray in aggregate. And I'm just showing this plot for brevity, but we've also confirmed this with differential abundance statistical testing as well. And these clusters could be grouped into established microglial states that have been found in the mouse and human brain. We can see a little interferon responsive cluster expressing MX1 and IFIT3, a proliferative state spanning multiple phases of the cell cycle, multiple antigen presentation states that differ depending on which substrate the cells were exposed to. And most interestingly to us, we found a microglial state that expressed markers of the disease-associated microglia, including LPL and GPNMB. So these states were mostly present when cells were exposed to either apoptotic neurons or recombinant amyloid beta. So we confirmed the existence of several of these states using orthogonal approaches. So for example, using GPNMB as a marker, we can show that apoptotic neurons increases GPNMB mRNA using qPCR and validate the same for protein using immunocytochemistry. And I should say we've performed similar experiments for all the other states identified in our data set and for also multiple other markers of this DAM-like state. So having generated these different microglial states in vitro, the obvious next question was to evaluate how similar are these states to those found in the human brain. So to test this, I leveraged a human brain data set from the lab generated from cortical biopsies of patients with either Alzheimer's disease or control, I should say. And I used data set integration to compare these two data sets, in particular an algorithm called Liger, which aligns data sets by identifying shared common gene signatures. And what we found was that human and IMGL cells clustered together really nicely, which is quite surprising, and only a small number of human cells clustered by themselves. And I want to be clear, this doesn't mean that the two data sets are exactly the same, but rather there are extensive shared gene signatures that group different subtypes together. So as an example, we were easily able to identify all the states I showed you earlier. So the top is IMGLs, the bottom is markers for data from the human brain. And so we can see clusters of DAMs, interferon-responsive microglia, and proliferating microglia as well. And in addition to this integration, I performed a battery of further computational analyses investigating the similarity of IMGL and in vivo microglial states. So I'm showing you one of these here, a gene set enrichment analysis performed on differentially expressed genes from the different human clusters against the IMGL clusters. And you can see hopefully that there's good correspondence, which is further evidence that these simple CNS substrates can induce microglial states analogous to those found in the human brain. And we again focused on a DAM-like state in vitro for further validation. So we found an extensive shared gene expression signature in both IMGL and human DAMs, shown here in red, as evidenced by their co-clustering, and you can see some of these signatures here. And all these genes were also upregulated in DAMs from one of Bart's papers as well. So in addition, we also confirmed that, like mouse and human DAMs in vivo, the formation of our in vitro DAMs is also dependent on TREM2 expression. So we can induce the expression of TREM2 with apoptotic neurons, but this is completely blocked in the absence. And we've done this for multiple different markers of DAMs as well. But I do want to highlight that the states identified in these two data sets, they're analogous, but they're not identical. I mean, these are cells in a dish, of course. And so to illustrate this, this is a comparison of differentially expressed genes from IMGLs in yellow and the brain biopsy data set in green. And you can see that although there's a large number of shared differentially expressed genes, there's also a lot more data set specific genes. So we view this as a big step forward in modeling human microglial states in a scalable way, but we still have more work to do to improve these in vitro models. So with the way of generating these human brain-like microglial states, we next focused on making these cells tractable. So microglia, as we probably all know, are notoriously difficult to genetically access using transfection or viral transduction. And as an example of this, when we delivered lentivirus to IMGLs and performed flow cytometry to detect positive cells, we got an incredibly disappointing result of only 4%, which, as you can all imagine, makes most experiments completely unfeasible. So we scoured the literature to see if there were additional tools we could use. And we found one study that discovered a viral protein called VPX, which could enhance lentiviral transduction in myeloid cells. and VPX degrades a human viral restriction factor, and we found the co-delivery of VPX with our lentivirus improves human microglia transduction from 4% to 90%. And so that really opens up these cells to facile genetic manipulations and large-scale functional experiments. So now that we can genetically transduce these cells, we're currently leveraging this to understand the functions of genes that define microglial states. And in particular, I'm focused on dissecting how microglial states are regulated by different transcription factors, with a particular focus on disease-associated microglia. Oh, sorry. So in order to identify regulators of in vitro microglial states, we combine two approaches. So first, I applied gene regulatory network analysis to our single-cell data, which takes into account both co-expression and transcription factor bindings data to nominate regulators of each microglial state. And secondly, we performed a TAC-seq on IMGLs that were exposed to apoptotic neurons or unperturbed. And this enabled the identification of open chromatin regions which correlate well with dam formation. And by intersecting the results of these two orthogonal analyses, we identified 13 transcription factors that we think are master regulators of the disease-associated microglia state. And this list includes MIDF, ATF3, EGR2, and STAT1, some of which we've heard about earlier. And we were particularly interested in MIDF because no studies had really examined this transcription factor in microglia functionally. And moreover, prior work from several groups had shown that MIDF was an upregulated transcription factor in microglia from Alzheimer's disease patients. And we found that MIDF was upregulated both in vivo dams on the left, in vitro dams on the left, and in vivo dams on the right in our brain biopsy cohort, which suggests that MIDF could be a dam regulator both in vitro and in human brain microglia. So to understand the function of MIDF, we took advantage of our new lentiviral approach to overexpress this transcription factor in IMGLs, or using mCherry as a control. And to examine the functional impact of MIDF overexpression, we used RNA-seq and phagocytosis assays. So even though we just overexpressed a single transcription factor, we were actually able to find hundreds of differentially expressed genes up and down regulated by MIDF in our bulk RNA-seq data. And a gene ontology analysis of MIDF-driven genes converged on lipid and in particular cholesterol biosynthesis metabolism, pathways which are also upregulated in AD patients. And digging a little bit further into the genes enriched by MIDF, we found that MIDF overexpression could drive the expression of GPNMB and LPL, two canonical DAM markers. And overall, we found that the overexpression could drive a small but significant subset of the DAM marker genes in IMGLs, suggesting that MIDF partially regulates the DAM gene signature. And finally, we wanted to know what are the functional consequences of MIDF overexpression. So to do this, we exposed IMGLs to our virus, transduced them with either mCherry or MIDF, and we exposed IMGLs to apoptotic neurons conjugated to a pH-sensitive dye. And we did this because increased phagocytosis is observed in many neurodegenerative diseases, and especially in disease-associated microglia. And strikingly, we found that overexpression of MIDF drove microglia into a highly phagocytic state where they engulfed a lot more apoptotic neurons than the mCherry control. And so collectively, these results suggest that MIDF is an important regulator of the neurodegenerative DAM signature and also drives increased phagocytic function. So currently, we're creating knockout lines for these iPSCs to target MIDF and also to investigate the other transcription factors nominated in our epigenetic analysis. So to summarize, I've shown you that exposure to simple brain substrates can induce stem cell-derived microglia to take on diverse transcriptional subtypes in vitro. I've shown that these in vitro subtypes are analogous to those found in the human brain. And I've also shown you that we've developed a novel viral approach to transduce human microglia in vitro. And finally, I've shown you how we've applied this new platform to study the regulation of the DAM state and identify MIDF as a critical regulator of DAM's gene signature and also phagocytic function. So together, we hope that this toolbox will enable the manipulation and functional interrogation of human microglial states in a scalable manner. And so with that, I just want to thank my PIs, Beth Stevens and Evan McCosco, my co-authors, Martin Therrien and Sashi Jarab, our key collaborators, and of course, funding. And thank you all for your attention, and I'm happy to answer any questions.
There's a question up the back there. Oh. Up the back first, and then there's second.
Sorry, thank you. Thank you very much for the talk. Yeah.
Oh, sorry.
Sorry.
I was wondering, because you mentioned that your DAM signature, if you like, in vitro, there was only a sort of partial overlap with the in vivo DAM of around 100 genes. And then comparing your MIT-F overexpressing iPSCs, or iPSC-derived microglia, to that in vitro DAM signature, you saw a certain amount of overlap again, but it looked like you were comparing that to that broader in vitro signature, which doesn't fully overlap with the in vivo DAM gene. So how much does your MIT-F-driven in vitro signature actually correlate with the in vivo DAM profile?
Yeah, that's a great question. It's about the same proportion, yeah. We were disappointed to see.
Over there.
Yeah, hi. So I was wondering if you've looked at what cell surface microglial receptors may signal down to MIT-F. You know, obvious ones like TREM2 or CLEC7A come to mind, which may already, we know, regulate the DAM phenotype. So do you plan to, or have you got any ideas of, you know, the receptor regulation down to these key hub genes?
Yeah, we're working on that in the moment, and we do see an effect of TREM2. So, yeah, more to come.
I have two questions.
Yeah, nice.
One of them is, why do you think not all the microglia in the dish respond to the DAM state, given the signal that all of them are receiving is the same? And then I'll ask my second question after that one. So
That's a great question, and we were really puzzled to see that, in fact, in some of our conditions, DAMs only make up 10% of the total number of microglia after a substrate exposure. So I think there's probably two reasons. So one is that we're looking at one time point, 24 hours, so maybe things start to converge later, and we do see a large population of kind of semi-DAM, somewhere in between, like in a dish homeostatic and DAM, that are kind of on their way to there, and we do see a clear trajectory. But the other thing could be some kind of epigenetic effect. So I think one of the things that we're doing right now, is trying to examine these at later time points to try and get at that question. But yeah, that really puzzled us as well.
intriguing, isn't it?
It is intriguing, yeah. And perhaps there's like epigenetic or some kind of cell-cell signaling that we just can't figure out by just doing single cell at one time point. So.
Mm-hmm. And my second question was, I'm really excited that you're able to, you know, overexpress a gene in microglia. And I just wondered whether you checked to see that they don't have a toll-like receptive viral nucleic acid response, because they will be phagocytosing and detecting whatever DNA you're putting in there.
Yeah, so it's a lentiviral system, and all viruses, including AAVs, which we think are inert, but especially lentiviruses, do induce an interferon signature, and we characterize this in our paper. What we see is if you use this virus and you kind of wait out the signature for about a week, the cells do return to a resting state, and then you can go and do your functional analyses. So we validated that with a bunch of markers and qPCRs at different time points.
I better read the paper.
No, I just...
Any more questions? Got one up there and then one down here.
I was wondering, for the knockout cell lines that you are trying to produce, you're using CRISPR or also lentivirus?
We're using both, actually. Yeah.
Sorry?
We're using both, actually.
Ah, so it also works for SH lentivirus. You're with the VIX system.
Yeah, so I think for more scalable experiments, of course, we want to be able to use the lentivirus, but for these experiments to kind of do them side by side as a validation, we're both knocking them down with lenti and creating CRISPR knockout lines.
Yeah, thank you.
Yeah.
Your beautiful work. So I was wondering, your MIT-F transduction, have you checked in your cells? Is that equally abundant in all your cells if you hit them, and does that maybe affect their proliferative capacity? Is that changed by the levels of MIT-F they have?
So we haven't done any single cell readouts with this yet, but we do see a whopping increase in MIT-F when in that MIT-F condition, I guess, which is good to see. We don't see any change in proliferation, but this is all at one time point. So I do wonder if we kind of wait a little bit longer.
Because you have an M cherry with there, right? So if you flow them, do you see that your cherry is that stable across all line cells? Or?
We use M-cherry as a control only, so we don't have mid-F M-cherry. We just have M-cherry and mid-F. So we use M-cherry as a validation that we can transduce the cells, and then we just read out the mid-F upregulation in bulk RNA-seq. But I agree, doing this single cell level and maybe some further assays definitely something of interest. Yeah.
Thanks.
Okay, well, one last question up there. Thanks.
Thank you. Yeah, I was just wondering if you've looked into the mechanism of the VPX VLP. So we use it too. So we use the Cowley protocol, and we transduce the cells at the sort of, as we plate them into the microglia media and then differentiate them for 14 days. But we found that when we tried to look into the mechanism, we found it was independent of its inhibition on SAM-HD1. Mm-hmm. So I was just wondering if you had sort of looked at how it might work, because we're a bit stumped about that at the moment.
No, we haven't actually looked into that at all. We kind of got it working and declared victory, so maybe we should have looked into it in more detail.
Well, it's not SAM-HD1, so...
My hope is that there will be some AAVs down the line that, from capsid evolution studies that will make, lentivirus doesn't, as people who use it know, like definitely drives a lot of, interferon signatures and stuff. Not that AAV is totally inert, but I think that'll help a lot. I know there are a lot of labs working on it.
Cool, thanks.
Well, thank you very much.
Thank you.
We'll move on to our final speaker of this session.